Dear students, in this lecture we will learn about morphology and the shell structure in Unio, which is a freshwater mussel. Now these freshwater mussels are found in most parts of the world, and you find them partly buried in the mud of rivers and lakes. Now Unio is found all over the world and in Indian rivers and other parts of the world. So they belong to the class, they belong to the phylum Mollusca. You all will be knowing by now, and you have already learned the general characters of mollusks and their classification. So these Unio, it belong to phylum Mollusca and class Pelecypoda. Now, as we said, these are the freshwater forms which are found in ponds, lakes, streams and rivers. And if you look at the movement of these animals, they are basically sedentary. Sedentary means they are not fixed to one particular point here, but they move very slowly. They just flow slowly through the mud or the sand with the help of its muscular foot. Now most of the time the shell will be properly closed, enclosing the soft body or the visceral mass, and it has got a muscular foot which will be protruded out from the shell when it needs to move from one point to the other, and it is only then the locomotion is observed. Otherwise it almost looks like a sedentary animal lying in one place. And then even when they are there lying on the sand, they don't burrow very deep, they are not they don't go deep into the sand or burrow and all that, because of the shape of its body. If you observe this shell that is shown on the screen, you can make out that towards your left hand side the area is little rounded, that is the anterior end of the shell, and then the other side is the posterior end, which is little tapering. And then it will have two siphons or tubes through which the water enters inside the shell. That is, we call it as incurrent siphon or inhalant siphon, and the other one is exhalant siphon, through which the water goes out. So since the body of the Unio is completely covered by shell, now how do you think this Unio will respire, or how does it eat? What does it feed on? How does the food enters into the shell? These are the doubts that comes in your mind. Now when we say there is an ingress of water or the incoming water, there will be a small siphon towards the posterior end through which the water enters into the shell. Okay, so this siphon is what we call as inhalant siphon. So the incoming water brings in the food particles for this Unio. Now what does this Unio feed on? They mainly feed on the microscopic organisms, both plants and animals, and they are basically called as filter feeding mechanism. They exhibit this filter feeding mechanism. So we will learn all these feeding aspects in detail when we come to or when we learn about the digestive system in Unio. For the time being, you know that the water that enters into the body of the Unio is the main source of food for Unio, and this water also gets in the oxygen through which the exchange of gases takes place. And the whole process of respiration, how it happens, there are so many complicated structures, or the gill filaments, the cilia, everything is involved. That we will learn in detail when we discuss about the respiratory system. Okay, now for this class we will mainly focus on the morphology or the external characteristics of Unio, and we will learn about the shell structure. How does this shell look like? Even when you take a cross section of the shell, what are the different layers that you find and how it appears. So we will mainly focus our discussion on the morphology of Unio and its shell structure. So shall we proceed? Yeah. Now, so here you can see the diagram showing the external morphology of the shell. So as I said, this rounded portion is what is called as umbo. Can you see here this rounded region here? This is called as umbo, and that is the oldest part of the shell. And it rests here, this side is the anterior one, which is little rounded, and this is the posterior end, which is slightly tapering compared to the other side. And then what else you observe? You observe n number of lines on the shell. So that is called as lines of growth. And we know that this Unio is a bivalve. Bi means two, valve. Why? What is valve is? Now this animal consists of two halves of the shell. Both the shells cover together, wherein it encloses all the visceral mass or the soft body of the organism. Since it is made up of two halves of the shell, we call it as bivalve. Okay, so when there are two halves, that means it needs to be tightly held together by some means, and it is this hinge ligament that helps both half of these shells to be in place. So we will learn in detail like what does hinge ligament do, and you have something called as hinge teeth and all that. So to have an overview, this is the morphology of the shell. And when you remove those shells, or when you take out both the shell halves, so each half of the shell is called as a valve. Okay, so when you just open both the valves, this is how the animal looks like. Now what you see inside is the visceral mass. Visceral mass means all the organs that are there inside the shell, or what is collectively we call it as visceral mass. Okay, now what all you see here? You can see that now this is one half of the shell. The other valve, we have opened it to see all this visceral structures, isn't it? So here this flap like thing that you see here with filaments here, those are the gills. Okay, and you have muscles which controls the movement of the foot, which is the foot here. Can you see this muscular portion? This visceral mass is called as the foot. Okay, so here you have two muscles, that is anterior adductor muscle and the retractor muscle. This is retractor muscle, helps in retracting the foot, and the anterior adductor muscle, and here it is the posterior adductor muscle. They hold this visceral mass in place. Okay, so if you have to open one half of the shell, you have to scrape out this muscle from the shell, then only it can be detached. So it is these adductor muscles which keeps this visceral mass in place. And then you have a fold of mantle. Now can you see here a layer of skin like structure? This is called the mantle, and you have mantle below this visceral mass, and there will also be one fold of mantle above this visceral mass that we have cut open along with the upper half of the shell. Understood? So if you go in a order, like for example, if you take this as a complete shell of a Unio, and if you go in a certain order, like you have the shell valve here, this represents the shell valve, right? This represents the shell. What do you find below it? Below it you find a mantle lobe, and then inside that you find this visceral mass, and below the visceral mass again you have a mantle fold followed by the lower half of the shell. Understood? So this is how the organism is, the different parts of this organism is arranged in Unio. Now we'll focus more on the structure of the shell. Now this Unio, the freshwater mussel, it is enclosed in a hard, calcareous, horny, blackish bivalve shell. Okay, it is hard because it is made up of calcium carbonate, hence we call it as a calcareous shell, and it has got horny kind of a texture. Horny means not horns, the texture, like it is very hard, right? So because of that we call it as an horny blackish bivalve shell. Why bivalve? Because it is made up of two halves of the shell. Understood? So it is made up of, or the muzzle is enclosed in a hard calcareous horny blackish bivalve shell, and the body of the Unio is laterally flattened. It is not round, it is not elongated, it is laterally flattened. The anterior side of the body is roughly oval in shape. You can see in the diagram the anterior portion is little oval in shape, and the posterior end, when it comes, it becomes little narrower, and it is bilaterally symmetrical. What is bilaterally symmetrical? You can divide this body into two equal halves as if it is a mirror image of the other, and the size varies from 5 to 10 centimeters in length. It can be of 5 centimeters, 6, 7, 8, 9 or 10 centimeter in length. And then when you cut open the animal, here as is shown in this second picture, the body is light cream in color. It will be very soft, elongate, oval with somewhat broad and a narrow posterior end. And then the mantle or the pallium, mantle is also called as pallium. It has two equal halves. The mantle lobes, that is, as I said, one lobe on the upper side and one on the ventral side. And then this one, like the line where the mantle ends, here if you see, this is the line that the mantle ends, isn't it? This is still this line, the mantle is present. By continuous presence of this mantle, one small mark will come at this margin, and that we call it as pallial line. Okay, so that pallial line is formed inside the shell, and the cavity that it encloses is what is called as a mantle or a pallial cavity. And then all this soft body of an organism which is present is what is called as visceral mass, or it is also called as a pallial complex. Now I was talking about to you regarding the siphons, isn't it? So these two siphons are the openings. One is dorsal exhalent siphon, like here you will find two openings like this. The dorsal one, which is towards the dorsal side, is the exhalent siphon. Why exhalent? It takes away the water from the animal. And then the other one will be the one which is lower to that. This is not the exact position because I am unable to draw very precisely on this. So the one which is below the dorsal siphon is the ventral inhalent siphon. Why it is called inhalent? Because it allows the ingress of water. For example, if this is dorsal and you think this is ventral siphon, okay, if this is ventral siphon, now here water goes out and through this the water comes in. Okay, so this is an inhalent siphon. But then how will you distinguish what is that key identifying character of whether it is an exhalent or an inhalent siphon? There is one more difference. The exhalent siphon will have a smooth outer margin, whereas the inhalent siphon, that will show a little fimbricated margin like this. I mean, if you see the outer surface of the, for example, if you consider this as the inhalent siphon through which the water enters, the margins will be little fimbricated like this. Whereas the exhalent siphon through which the water goes out, the siphon will be smooth without any kind of fringes. Okay, so that's the difference, and that's how you can identify whether it is a exhalent siphon or the inhalent siphon. And then when the animal is in rest, like when the animal is not moving, you will not come across the formation, you don't see this muscular foot outside. That will be enclosed within the animal and it will be inside the shell only. So in addition to that, in this visceral mass, as we have already discussed, you will see the gills, or gills are also called as ctenidia, and then you will see the renal pores and anus and the whole part of all the visceral mass, which includes the digestive system, circulatory system, excretory system, respiration. Everything is put in the form of a mass, and which forms the visceral mass, which is enclosed by this shell. Now, when you look at only the shell now, here it is clearly shown the upper part of the shell, which is little rounded, is what is called as umbo, and this umbo is the oldest part of the shell. That is, when a young Unio is formed, the first ring of shell, when it forms, that gives rise to umbo. So when we say that is the first formed formed part of the shell, so when you consider an adult, that will be the oldest one, isn't it? So umbo is the oldest part of the shell and it is first formed in the young one. Now, what do you think that this valves or the shell is made up of? We said they are two equal halves. They both are equal in size, so that no part of the animal is protruded out. If you see in some oysters, what will happen? The lower one will be little smaller and the dorsal one will be little bigger. There could be unequal shells also. But in Unio, both half of the shells are even. That means they both are equal in size. Okay. And this is made up of a substance called as conchiolin. So this you should remember. This can come as a one marker question. So the shell is basically made up of a substance called as conchiolin. In addition to that, it is also made of the salts of calcium carbonate. We call it as calcic substances. Okay. And it grows along the margins with the growth of the muscle. As the muscle ages, we said this is the oldest part of the shell. That means after that this would have come, then this would have come, then this would have come. So it grows along its margin, and the last line will be the youngest or the recently formed lines on the shell. Now these lines that we see on the shell is what is called as lines of growth. Okay, you call these as lines of growth. Now why we call it as lines of growth? As and when the animal grows, it adds on each line to the surface of the shell like this. Okay. Now people say that by looking at the arrangement of these lines, you can say like whether the animal was subjected always to the continuous supply of food, or whether it has experienced any kind of drought or lack of food. How means, see in some you can see the lines will be very closer like this. Okay. So when the lines are closer, it shows that plenty of availability of food. The food was sufficiently available, so the animal could grow in a same pace. Now after this line, you don't find any other line till here. There is a gap of this much. Okay. That means from this period to this period, there was lack of food. Because of that, the growth of this animal was shunted. So only once the favorable conditions prevailed, then only again the growth of this organism took place. Okay, so that is one thing that you can know from this lines of growth. So these lines we call it as lines of growth. Now we said the shell has two valves, and these two valves are hinged along a straight dorsal hinge by a tough elastic hinge ligament. I hope you understand what is a ligament. It is this elastic kind of a substance that holds this both the halves of the shell tightly so that they do not slip away. And each valve bears a teeth-like projections, and these teeth-like projections are called as hinge teeth. They are present along the hinge line, and this teeth of one valve fit with the socket of the other, so that whenever the shell opens and closes, there is no chance of displacement of these shells. Understood? So you find, as I've already told you, you find series of concentric rings, the lines of growth widening towards the margin are present on the outer surface of the valve. And then we spoke about umbo, that is the oldest part of the shell, and the inner surface of the shell. So this is with respect to the outer surface of the shell. When you look into its inner surface, the shell is very smooth and it has pearly appearance. I hope all of you must have seen pearls, and you know that pearls have got some kind of luster. That shining nature of pearls will be there, right? So this inside the shell, if you see, this is how it appears, like a pearly color with faint bluish tinge. The figure B here shows the inner surface of the shell. And then I said that the inside of the shell will be marked by a very thin line where the mantle fold ends, and that line is what is called as a pallial line, which marks the outer border of the mantle lobe, and it is present towards the inner surface of the shell, very close to the margin. So these are the things that you see when you observe the external surface of the shell and internal surface of the shell. If you are asked to write or describe the shell in Unio, you have to write both external view and internal view. So you will discuss or you will describe the umbo, the lines of growth, the hinge ligament, the hinge teeth, and then inside when you come, you will write about the muscles. Can you see two scars here? I will discuss further regarding the muscle scar. In this picture also you can see these two patches, which are nothing but the scar of the muscle, from where the muscles has been detached. So you will write about the anterior adductor muscle, posterior adductor muscle, then retractor muscles and the protractor muscle also. And you should not miss out on the pallial line. So any of these terms can be asked as 1 mark question. So you should be thorough and you should know what it is. Now this picture gives you a clear idea of what we have discussed so far. So this diagram A shows you the complete animal of Unio with hinge line, exhalent siphon, inhalent siphon. Here you observe exhalent and inhalent siphon. We said inhalent siphon will have little fringe like fimbricated margin, whereas the exhalent siphon is shown as a smooth curvature. Sorry. And then here in the picture B, both halves of the shell has been cut open. They have been opened up to show the visceral mass, the attachment of this anterior adductor muscle, posterior adductor muscle. These muscles have been scraped off from the these muscles have been scraped off from the upper half of the shell, so it leaves a scar there, and here you can clearly see the pallial line, which is represented by the outer margin of the mantle, and foot is clearly shown how it is enclosed within the shell. Only when the animal has to move, that foot will be protruded out with the help of this protractor muscle. So I hope all these things that we have discussed so far is clear to you. Moving on, it is the same diagram. This diagram you have to learn in addition to the external view, and all this labeling should be there, and it can come as a 5-marker question. So anterodorsal scars towards the anterior and the dorsal side. You have the anterior adductor muscle, okay, which is slightly large and it is round, and this, and also you find the I hope you can see this. This is posterior adductor muscle. Here the one which is little large and round is the anterior adductor muscle, and then anterior retractor muscle are also found. Where is it found? This is slightly smaller, and this is towards the posterior dorsal side to the adductor. You should not get confused. You can just know that it lies above the anterior adductor muscle here. And then you also have the scar of this anterior protractor muscle, okay. Whereas in the posterior dorsal scars you have posterior adductor muscles which is large and oval, whereas anterior one is round, the posterior one is little oval in shape and large, whereas the posterior retractor muscle which is small and close to posterior adductor muscle. Now if you see these adductor muscles, what do? The adductor muscles, they help in closing the shell valves tightly by pulling them together. Hence they are attached to both halves of the shell, so they pull them together, whereas the retractors pull in the foot. And what does the protractor muscle do? Whenever the animal has a signal to move, this protractor muscles pushes the foot out, and the retractor muscles retract the foot back into the shell, and these adductor muscles, they hold both halves of the shell very tightly, hence they are pretty large and strong as well. And the hinge ligament, it acts antagonistically to the adductor muscles and causes the shell valves to open when the adductors relax. When these adductor muscles relaxes, the hinge ligaments get pulled up and hence the shell opens. When these adductor muscles contract, the hinge ligament should relax so that it facilitates the closure of the shell. So they both work antagonistically, opposite to each other, hence facilitating the open and closing of the shell. So far what we have discussed is regarding the morphology or the external features of the shell in case of Unio. And the next thing you need to learn here is the structure of the shell in a sectional view. Like when we said while discussing about the inner surface of the shell, we said it appears pearly in nature from inside, and from outside it appears little horny, blackish, dark pigmented one. So if you take a section of it, like what are the different layers that it is made up of, what are the substances that contribute to it, that you will learn in this small topic. So once you take a section of the shell, you come across 3 layers. One is the outer periostracum, and the middle prismatic layer, and the inner nacreous layer. So all these layers are followed by the mantle, because we said inside the shell is in direct contact with the mantle, both on the dorsal side and on the ventral side, isn't it? So shell basically shows 3 layers: periostracum, prismatic layer and nacreous layer. This can be asked as a 3-marker question. Name 3 layers of the shell, then you should be writing periostracum, prismatic layer and nacreous layer. Now let us understand like what these layers are made up of and what makes this shell appear so shiny from inside. Now you have to practice this diagram. This is the one which is there in the textbook. So if you are asked to write about the describe the sectional view of the shell in Unio, you will draw this diagram and write about all the 3 layers. This will fetch you 5 marks. Now the shell, it consists of 3 distinct layers. The outer periostracum, inside the middle prismatic layer, and the inner one, the nacreous layer. This periostracum is the thin outermost layer which is made up of conchiolin. Have you heard about this term? Yes, we said the shell is made up of conchiolin, isn't it? So since this is the outermost part of the shell, obviously that is also made up of conchiolin, which is a chitin-like substance, and it imparts color to the shell. Okay. Whereas the middle prismatic layer, which is also called as ostracum, this prismatic layer is a thick middle layer. It is composed of alternate layers of conchiolin and the prisms of calcium carbonate. I hope you understand what we mean by prisms. That means the crystals of calcium carbonate. Okay, so even if you observe the diagram clearly, it is represented in 2 alternating layers. Now what does this alternating layers shows you or tells you? One layer is made up of conchiolin, as you see in periostracum. It is represented with the same color and the texture, and the line thin that is there represents the crystals or the prisms of calcium carbonate. So here it is made up of 2 different substances which alternates with each other. One is the conchiolin layer and the other one is the prisms of calcic substances or the calcium carbonate crystals. Then the innermost layer, that is the nacreous layer, is what is called as mother of pearl. So any guesses why it is called mother of pearl? Because it is this layer that secretes the formation of a pearl. You know how these pearls are formed? I'll come back to that later. First let me finish describing this layer. So this layer is called as mother of pearl. Now this is the innermost layer, which is composed of alternate layers of conchiolin and the calcium carbonate crystals, and the layer is smooth and lustrous. I hope you understand what is luster. Luster means the shining, the shine that the pearl exhibits, that is called as lustrous. So this periostracum and the prismatic layer, they are secreted by the mantle edge, whereas the soft and horny in color at the beginning, which hardens with subsequent deposition of materials. Okay, I hope you understood what I meant. Since this nacreous layer is secreted by the mantle and it is close to the mantle, it is towards the inner side. So it takes only the conchiolin and the calcium carbonate crystals. Whereas prismatic and the periostracum, if you look at, why it becomes hard and darkly colored is because that get exposed to different kinds of material that are there in its environment. So when all those salts come and deposit on that layer, it becomes darkly colored and it becomes little hard in texture as well. Okay, so this nacreous layer is secreted by the outer surface of the mantle, and the pearl is secreted by this layer of nacre. Okay, hence it is called as mother of pearl. I hope this is clear to you, and I'll show you this interesting picture which shows the freshwater mussel. Both half of the shell is wide open. You can see the visceral mass, and the very interesting thing that you see here is the formation of pearls on either side. Can you notice that? So this is the shell, this is the mantle, and we said it is this mantle that secretes this nacreous layer. So and from that nacreous when the mantle secrete those nacre, which is the mother of pearl, that give rise to the formation of these pearls. Now the question is, how does this pearl formation takes place? I'll tell you, it's very interesting. See, we said there are two siphons, the inhalent siphon and the exhalent siphon, and we also said that Unio is a filter feeding, it exhibits filter feeding mechanism. That means it filters off all larger particles when it tries to enter into its body. So by mistake, when some dust particles or something enters into the Unio, and if it gets trapped in this mantle, that irritates the animal, because this is the body of the Unio, which is very soft, isn't it? So when some dust particle or a little larger particle comes and gets entrapped in this mantle, that irritates the animal, and because of that irritation, this mantle starts secreting more of nacre. Okay, so we said nacre is the mother of pearl, isn't it? So it is this nacreous layer that secrete pearl. So when the animal gets irritated, this mantle secretes more amount of nacre, and so that it can get rid of the irritation. So this nacre, when it gets secreted more around this dust particle, then it results in the formation of pearl. So that's how pearls are formed. You must have heard that the original pearls, and once the pearl is very much exact round in shape with that beautiful luster that costs some millions of dollars, and the queen will wear it and all that story will be coming. So that's how original pearls are formed. Now what you see in market is, you see the plastic pearls also, which is just made into round shape, and they put on some kind of paint so that it appears luster. And if you have purchased original pearls anytime, then you know the cost of it ranges in several thousands. So you have a specific industry also called as pearl industry. What do? They rear the oyster, they induce the dust particles inside these animals and live, so that they can form pearls. So if you inject or if you introduce an elliptical shaped dust particle, nacre will be secreted around it, which results in elliptical pearls. And if you put some circular kind of a larger particle, you get a circular pearl. So it is like that. So people rear these freshwater mussels or even few oysters to rear pearls. So I hope I have made these two things clear to you, and all the structures and the morphology of Unio and the structure of the shell. I hope you have enjoyed listening to this lecture. Thank you very much. So if you have any doubts, or if you need any more clarification, you can mail me at this email address, vlatadamodaran@gmail.com. So if you like this lecture, you like it, and subscribe to my channel, and also you can give your valuable comments, which will be highly appreciated. Thank you.